Well, we thought that we'd spice this up a little bit more than before lunch to try to be three people on stage. So see how that goes. So after lunch now, we are still going to talk about how to facilitate structure-based design. But today, well, on this presentation, we're going to focus on how Plexus Design can help us at Sprint Bioscience to do this. So we're still in the business of making better molecules faster. For all of you that were here before lunch and paid attention, there will be a couple of slides that will be repetition, but I'll try to keep it short. So Sprint Bioscience, we're still a drug discovery company focusing on treating cancer. We work in the preclinical phase. We use fragment-based screening and structure-based design to move our projects forward. And we do that all the way to clinical testing. And the focus of today's talk is around this box, medicinal chemistry, that we call it because we're chemists. So driving the medicinal chemistry plans, once again, this is truly crucial, and that's why it's worth taking the time to say it again. The design part, we want to make a better molecule. We need to know what do we want to improve. What is the hypothesis? That's key. So to make the design, make the compound, test it, analyze, and go back. And to make this cycle go faster and to make it pop out even better ideas and better molecules. That's why Sprint Bioscience is really good. So once again, what happens? Idea to design to target molecule. We still have an hypothesis. We want to populate that hypothesis with ideas, with molecules. Depending on what type of question we have, we will populate the idea in different ways. Can we build into this pocket? What types of interactions can we make if we want to improve potency, for example? During the Marvin Live talk, we focused on few structure designs. But now, looking at the populate part, we want to know what types of structures we want to populate with, but also how many structures can we assess. When it comes to evaluate, we have the power in the computational part of the medicinal chemistry team to evaluate lots and lots of molecules. So if the idea can be populated with a lot of structures, the chance is that we will find a better idea, or many ideas, so we can be picky. The synthetic chemist can actually select the most feasible synthesis to make the best compound. So once again, how to make this process better with respect to populating your ideas with many molecules. What we didn't have in the company, well, we said that how can we do this better? What are we lacking? So Marvin Live was one thing. To get access to chemical space for everyone in the company, once again, you have the computational chemists can enumerate virtual libraries easily. But for the rest of us, if you have an idea and you want to populate that idea with structures, you could write on a piece of paper, and after 4 structures, you get tired, that's it. Maybe you write a comment, add stuff here, and they give it to the computational chemist. Or you go into a sketcher, and you can copy-paste, but still, after 10, 15 compounds, you get tired. And you hope that the idea that you have still comes through in those few molecules that you actually make. So of course, being able to use the machines to enumerate is great. So that was the computational chemist, but also for the medicinal chemist or the synthetic chemist, actually having a tool that is easy to use to do this. That will make people use enumeration, use the machine to actually expand chemical space. And that will increase the diversity of the molecules that we will evaluate. And increase the chance of finding something that is really good, and we make it and bring the project forward. And the third part that we wanted to improve, once again, to improve the workflow when sharing and documenting and evaluating ideas. Once again, a lot of post-its, maybe with a scribbled idea with R groups and put hetero arrows here, maybe something with a donor in this and this position. If you have an improved workflow where the ideas can actually be documented, you can write comments, that would really improve the way that idea generation is handled, and we can faster go to the lab and make the molecules. So these were the things that we really want to improve. So how come we chose Plexus Design to do this? And to explain that, Anna, please show us what Plexus Design actually can do.
So Plexus Design is a tool that can contribute, that can give a valuable contribution to this process based on ChemAxon technology and also on the computational power. This tool allows you to generate new ideas and create large virtual libraries, but this is only the starting point. Of course, we need to provide additional solutions for post-processing of the data, and I have selected a couple of post-processing options or opportunities we offer. You can characterize your compounds, calculate physicochemical properties, and do filtering and searching based on those properties. You can perform structural-based searches. You can send molecules to similarity search and perform similarity search against even millions of structures. You can register your virtual compounds to a central database, or, of course, you can brainstorm about the molecule or molecules of interest. You can enumerate large libraries with two different approaches. One of them is the so-called scaffold-based enumeration. Here, you have to provide a core structure. This is an example of a scaffold with variable parts. You have to provide the fragment definitions. These R groups represents the variable part. And then, using our Markov technology, this will systematically connect the fragments to a core structure and provide the large library. This is a pure combinatorial approach. The other method is the so-called reaction-based enumeration. Here we need a properly designed reaction. This is also just an example, which takes chemistry into account. You need to set the reagents, and then the software will eventually produce the library for you. But how can it be used in a workflow? What's the final goal of using this enumeration tool? What's your mission?
The mission. Yeah, if you remember from the Marvin Live presentation, our mission then was to scaffold hop, to create a new series based on another. So now we have a different mission. As Fredrik was saying, we are working with fragment-based drug design. So here our starting point is a small fragment, again crystallized in the active site of the protein. And our mission is to create a lead compound from this fragment. And usually that means that we increase the size of the compound by 3 times, and we need to increase the affinity of the compound by 1,000 times to the protein. So that's the mission. We need to grow our fragment, we need to expand it. And then the first thing is to decide where do we want to expand this fragment. And that's a completely different story. So we will just tell you that we want to expand it in 4 different directions. Each direction is represented by an arrow, and 1 arrow means 1 virtual library. We can create this virtual library using Plexus, or I can use the Schrödinger enumeration tool. Usually, the virtual library contains about 100,000 compounds. These compounds are then evaluated in the Schrödinger tool that is super good for evaluating ideas. I love the dockings. I love the computational stuff in Schrödinger. Very good. And also, or usually before we do the Schrödinger stuff, we often do some pre-filtering on phys-chem properties. So in this plot here below, you can see that for 5 of our 3 series, we actually have a strong correlation between lipophilicity and clearance. So we want to remove all virtual compounds with a log P below 2.5. We also want to remove all compounds below a molecular weight of 450. So we have the glide dockings, the Schrödinger filters, we have the filters on phys-chem properties. And then by the end of the day, we usually have 5 compounds that we actually synthesize. So these filters are reducing our compounds from 400,000 to 5 that we actually decide to make. So now Anna will show you how to filter in Plexus.
So how does Plexus work? I have selected some features to demonstrate you and give you some flavor of the application. This will be a demo video, so you can start the enumeration from predefined templates. Simply send this template to the Plexus Design tool, and everything will be set. So the scaffold, all the fragment definitions, we have the preview down there, and also the estimated library size. Then you hit the enumeration, and this will direct you to the resulting page, which will contain the enumeration result in a spreadsheet-like fashion with all the details. You can even rename the table if you wish, and then later, of course, you can filter the data based on properties, which first you need to calculate. There is an icon, property calculation icon on the toolbar. You hit the icon and the drawer opens. This contains all the available physicochemical properties. You select those ones which will be interesting for your further filtering. Hit the add button, and this will be calculated immediately, and your spreadsheet will be expanded with these calculations. And then, of course, later on you can do structure searches combined with non-structural data search and filter for log-P and so on. What else can you do with the tool? You can also select some compounds and, for example, explore the property space of that particular compound. Here you can see all of the available calculations we have. You can even specify which ones would you like to see on the screen and focus only those on which are relevant for your work. You can even customize these, the settings of these calculations, change them, update them, and then analyze, for example, the solubility of a given structure. Or if the details of the structure are way too small, you can, for example, enlarge the molecule. It's easier it's easier to see the details what's on the widget. You can also send molecules, as I mentioned before, to our similarity search engine, which was introduced yesterday. Simply right-click on the structure, hit the similarity, and this will direct you to this MedFast search engine, which provides you a real-time feedback about the similar structures compared to the one which is drawn in the Marvin.js. And then if you have some special compound of interest, you can select them, and of course you can export them. These were just some, as I mentioned, selected features. But how can this tool be used in a workflow? How does it fit into a workflow? Would you please explain that?
Sure. Well, how it fits into a workflow at Sprint Bioscience. Once again, the mission, what we want to do, taking a fragment hit to a lead compound. So expanding the ligand or the fragment in different directions. So the first case study is that I have an idea. Let's do something like this. So that's the fragment in the beginning. Looking at the crystal structure, we know that this area up here looks like it's going to the selectivity pocket. So the R group here could be aryl, heteroaryl, cyclic. It could be, well, could we maybe have a linker, a carbon linker, an oxygen linker, something. The small part here, it looks like something could fit there, maybe a 6-membered ring, 5-membered ring. That's my idea. Now let's start working on it. And then, but how to make it? I have no idea. What reagents to use? I have no idea. I don't care. I just want to create diverse structures. I, as a synthetic chemist, want to make a few. I have an idea. As a computational chemist, I might want to make 100,000. So I go to the scaffold-based enumeration tool. So we want to keep the morphine intact. The R group here is just what I, as a synthetic chemist, would probably draw 5 or 6 or 7. and then the R group here, the one that goes up here. I can draw them, I can import them from files, or if you have many, if you want to use many, then you have to have predefined files. But otherwise, it's so easy to just draw them, and you enumerate, as Anna showed, and you get your very diverse set of compounds. Easily, even as a synthetic chemist, you can easily generate 100 compounds instead of the 4 or 5 that you would manage by hand. So extremely easy to use. The diversity you get in there is actually more than you had hoped for in the beginning. Maybe you get combinations that you haven't thought about, but those could actually take the project forward. And also, the result is in an SD format, so I can get an SD file to the computational chemist. So using scaffold-based enumeration, at least for me, I open it, it's so easy to use, I get really happy. We talked about the wow factor before, this is wow factor for me. I'm very happy. So the second case then, moving ahead a little bit in the design, we made a couple of compounds. We've decided on the hinge binding motif and also the actual scaffold. So what we want to do now is up in the selectivity pocket. We know basically that what we want up there. We want an aryl group. But let's expand. We want to make a huge expansion. Once again, you can take the medicinal chemistry hat or the synthetic chemistry hat. But now I know I'm going to, in the last step, in the diversity introducing step, I'm going to use Suzuki reaction. So I want boronic acids and boronic esters. If as a synthetic chemist, as a medicinal chemist, as a computational chemist, you want to make a huge heat explosion, you take a large set. You might have all commercially available boronic acid and esters from Molport, for example. You take that. Or you might have virtual ones. You don't really care. You just generate it yourself. Or as a synthetic chemist, you might want to be a little bit more focused, a little bit more down to earth. I want commercial ones that I know that I can buy. I go to Molport, maybe. Or the ones that we have in-house. I have them on my shelf, the available ones. So then, reaction-based enumeration. You can draw your reactions yourself, or you go to the library reactions and just plug in Suzuki. You draw your, in this case, the core structure here is actually the R2, and then the boronic acids. You have that in a file. I just go to our reagent database and export the ones that I like, click enumerate, and it's there. Once again, easy to use, both for large R sets from the database, or you can even draw the ones that I know that I have these 5. These are my favorite boronic acids. I know them by heart. I just draw them very quickly. And once again, the results in an SD format, which makes the follow-up evaluation so much easier. So, what's good and what do we need? What do we want to improve? Once again, dream mode.
But first, good mode. So what is good? We think that Plexus Design is good because it's easy to use for all of us, not only the synthetic or medicinal chemists, but also I get happy from a tool that is easier to use than the Schrödinger version. So since I admit I'm a bit lazy person, after 4 or 5 scaffolds drawing in the Schrödinger tool, I get tired, and when I start using the Plexus Design tool, maybe I can draw 8 cores without getting tired. So we think that will be better molecules out of it, since computational power can handle 800,000 compounds or 400,000 compounds. And since the Schrödinger tool is really good to fish out the good compounds from those huge, large virtual sets, we think we can do better. The more, the merrier. And also, since we have a better workflow, we will save time. We will work faster. Time is money, if you're management. For me, time is happiness. Every second I can reduce doing tedious or boring or repetitive stuff is time that makes me happier. So we like that. We have some enhancement suggestions. I think it would be quite powerful to be able to filter your virtual libraries on the fly. So usually we don't want to see compounds that get bigger than a molecular weight of 450. We don't want to save or create compounds with a lipophilicity higher than 2.5. So I think that would help us make even bigger libraries, but you just get rid of the rubbish. Also, if it would be possible to improve the spreadsheet functions in Plexus, I think that would be powerful. Make it even easier to use when you're working in the spreadsheet function. Small thing, being able to strip salts from your reagents. You can do that if you have a KNIME workflow, but if you don't, maybe it would be nice to include it. And also for me, as a computational chemist, I think the naming of the products, the products that are enumerated, if I get good, reasonable names automatically, for instance, including the reagent IDs from all the reagents that I have created my library from, that would be really powerful. And also unique IDs. So if I have enantiomers or tautomers, I want to distinguish between all of them so that we pick the unique best compound from the enumeration. That would be nice. Also, we would like to wrap up by comparing Marvin Live and Plexus Design. So we talked previously about Marvin Live before lunch. And we think that both of these tools will help us enhance and improve both of the two superpowers behind molecular design. So when you're doing drug design, you have the human brain and you have the computer. And those are really the superpowers that we want to help do better. So we think that Marvin Live represents helping the human brain to create better ideas. As a human being, when you're creating ideas, you automatically take into account everything you know. So the SAR for your project, your previous experiences, your synthetic knowledge, what worked before, what didn't. And that's really powerful. So when you're creating ideas just using the human brain, usually the ideas are quite good. So we want more of those human ideas. And then the superpower of the computer is the ability to filter huge libraries. And we like that as well. So the more the merrier. The more ideas we put into the computer, the larger the chance that the super filters from Schrödinger will pick out something good. And again, I mean, there's a human being behind the enumerator as well. So if you put in some stupid enumerations, you will get stupid out. But that's a different story. So with that, we hope that we can do better and faster towards our final goal of treating cancer.
Basically, besides the fact that we provide a tool that can generate new ideas, we also find it important to connect the Plexus Design to additional tools in order to cover more and more workflows. And also, we are open to hear new ideas and new needs in order to make this tool better and meet the end user needs. This is integrable. So Plexus Design is already integrated into Stadrup and also into Live Design. And last but not at least, we really appreciate all the feedback and the continuous cooperation we receive from you guys. And then I think this is this describes very well the collaborative nature of both companies. So thank you very much for you guys, and thank you for your attention. Do you have anything to add?
Yeah, I remember the goal that we had, what we were trying to actually increase at Sprint Bioscience with going to enumerator. And now with the Plexus Design, to be able to expand chemical space is actually available for everyone in the MedChem team. Even if you don't want to care about SD files containing boronic acids or amines, you can just go in and use the scaffold-based enumerator and really expand a chemical space with more diversity than you originally had in your idea. Easy to use, happiness, we talked about that, and also then the workflow, once again, getting a little bit more organized when it comes to files and where to keep everything. So it's something that's helping us and hopefully will help us even more in the future. So, thanks.